Welcome to the third Forerunner series by Sci-ROI. So as you're already aware, Forerunner series aims to talk of established scientists who have transitioned to India. So getting to know of their challenges, experiences, and outlook will boost the spirit of the budding young scientist who would want to come back to India. Today, as part of the Forerunners, we have with us Professor Umesh Varshney, who has over 3 decades of experience in running his laboratory at Indian Institute of Science, Bangalore. Professor Varshney completed his master's from B.G. Pant University of Agriculture and Technology in 1979 and moved to Canada to pursue his doctoral studies from University of Calgary in 1985 and postdoctoral studies from Massachusetts Institute of Technology before moving to India in 1991 as assistant professor in IISc. Professor Varshney's research focuses on mechanistic aspects of protein synthesis and DNA repair using E. coli and mycobacteria as model organism. He is a J.C. Bose National Fellow of Department of Science and Technology, an elected Fellow of Indian Academy of Sciences, Indian National Science Academy, and a recipient of Shanti Swarup Bhatnagar Award for Science and Technology, one of the highest Indian science awards in 2001 by CSIR. In addition, he has also received G.N. Ramachandran Gold Medal for Excellence in Biological Science and Technology for his contribution to biological sciences in 2014. On behalf of Sci-ROI, I welcome you, sir. Welcome, sir.
Thank you. Thank you.
Okay. So today we would start with asking you, why were you really excited to do science? And what is the reason behind going into research?
All right, that's a bit tricky question right now. Let me put it in a bit of a context. So I grew up in a village, and the times that we are talking about is, late 60s and early 70s when I did my schooling. And these are the days when you will know that even calculators were not available. Yeah.
Yeah.
And so much of the motivation or inspiration that would come would be from the teachers that actually teach you in different classes, the way they teach. And not all the time people are lucky to have good teachers. I've been lucky that, many of the teachers that taught me were actually quite motivating in, they taught well. They sort of, raise the enthusiasm of doing things. Now you should sort of, in those days, and particularly in a village setting, the only aim a student has really is to do well in the exams and get good marks.
True.
Yeah. So then how does one, sort of, get into science? And that's a difficult thing. But, one good thing that happened in the college that I was studying, this is, the intermediate colleges are called, up to +2, they're still called as colleges there. They used to have these practicals for 9th, 10th, 11th, 12th standards. And unfortunately, I don't know whether we are doing those enough these days. And they will, teach you small things, if you think about, chemistry practicals, for example, how to identify carbonate, how to identify different, compounds and, groups that you have in a mixture and in pure form. That sort of raises some kind of curiosity. And so that's the kind of a beginning, I think, of how to really think about bigger things there. And I sort of, I think, got a little bit more intrigued by these things. I even actually set up very, very preliminary kind of laboratory in my home to do some of these things. So I think that's really what has been the motivation and the beginning of how to sort of, think about science and how to do things that are unknown. And in fact, I think there, if your teacher and books cannot tell you things, everything is unknown because there's no library, there are no journals,
Internet.
Uh, no internet, calculators were not there. Yeah. So
Yeah.
I think if a teacher can get you to think about evolution, which they did, by the way, because when they are teaching taxonomy, they are asking you to make herbarium, they're asking to make a collection of different animals, for display in their practicals, which I did very religiously. Really, I did more than what they asked for, and I think that must be the foundation for what really led me to get into science. I'm sure there are many other things, but it's a cumulative effect of the entire environment, and I think the initial education, which has impacted, I think, how to take my studies forward in science.
I see. So, so with that curiosity as your foundation, then later on you moved to University of Calgary to pursue.
Yeah,
Eventually moved
actually,
to.
I should say that before that I was at Panagar,
Mhm.
and Panagar, it is interesting that you are interviewing me, you're at Illinois, had a lot of collaboration with Illinois University.
I
And
see.
so much of the education at Panagar in those days was really very, very similar to what one would have studied at Illinois, because all the
Mhm.
course programs, the trimester system, I don't know whether in Illinois they're still continuing with trimester system. In Panagar, they have done away with it now. They've gone into semester system. So that was a very good foundation, in the sense that uh, they had the course system. You take a course with certain credits and you study that, and so one can make up for the deficiencies by taking extra courses.
Mhm.
And so that, I think, was also important chapter in my life, really. uh,
Okay.
I think the two major places where my foundation was built really was up to +2 at in my village, and then coming to Panagar to do my masters.
Okay.
I see.
That is also a place where I have really been motivated a lot by some of the teachers, very fantastic teachers.
Okay. So, uh, so Pantnagar, sorry. The GB Pant University is where you got your exposure
Master,
to, let's say, the uh, world leading education.
I would say so, yes. Yeah, that was
Correct.
the place where I think I got the introduction to what I don't know.
Mhm.
There was so much exposure at that point. Uh, Panagar University had a fantastic programs. They would bring the books of the authors that would not be available in the market, and they will actually give you 50% discount on if you bought them as a
Oh
personal
wow,
Oh,
copy.
that's so nice.
that's
That
very
was
good. Yeah,
another
for
Exactly.
the poor student. Yeah.
thing that
Yeah.
you could own your books, which you wouldn't have access to.
Yeah. Yeah.
Yeah.
Yeah. That was very nice. And okay. So, uh, and then after getting all this education in India,
Yeah,
uh, and then you moved to University of Calgary
that's right.
in
Yeah,
Canada.
yeah,
And then you pursued your PhD in molecular biology and biochemistry.
that's right.
So, how was that experience like? Why did you first decide to go to Calgary
That's
and then?
yeah, that's another interesting story, really.
Mhm.
So in my time, after doing masters, people used to get jobs in this agriculture services. Uh, ARS, I think agricultural research services.
Yeah.
They used to pay something like 750 rupees a month salary, and that was considered as a very good salary those days
Mm.
for a job.
Yeah.
And so there was inclination to do that.
Mm.
Uh, but there was also this, as I mentioned, that Panagar University had a good library,
Mm.
and often, I mean, of course, we'll get only one copy of Nature, Science, or whatever journals you have. But generally, there was this, culture of going and sitting in the library and reading through the nature. Of course, these days, if we can read through the titles of the journal, that is more than what one can ask for. But those days, I think, we were at least reading the abstracts of all the articles that were there in the particular issue of Nature. And I really never had thought of Calgary, never knew actually about Calgary. But when you flip through towards the end of the magazine or the journal, you would find a lot of these advertisements for vacancies and scholarships, all sorts of things. And this University of Calgary advertisement caught my attention because this is one thing, sometimes money helps. They were paying a scholarship for doing PhD, which was in the range of $800 in those days.
My God.
And so that was, I said, okay, rather than doing a job, why not do a PhD and also get paid well? So that was really, I would say it's a freak incidence that took me to Calgary, really. I applied there. They selected me. Now, of course, I didn't have the passport. But I went to the Canadian High Commission. They pretty much gave me everything without a passport. And when I got the passport, then I just came out of there. So I think, yeah, this is quite interesting thing. I never planned to come to Calgary, never planned to come anywhere to US or Canada, but it just so happened.
I see.
Yeah.
And I guess, yeah, so basically serendipity led you to Calgary.
Actually,
yeah.
This is really something I keep thinking about. And all these years, I used to get surprised that, everything just sort of happens for me. But then, I came in touch with Khurana when I was at MIT, got to know him. And he wrote an article in Science, and some of the opening lines of that article were very, very, inspiring. He also did something, he said that he never planned his life, but he did the best, whatever he got the opportunity to do for. And I think that is really what I think one needs to think about, that whatever opportunity you get, I guess you try to do the best you can at that place. And I believe things will then follow.
That's great, sir. That's great to know. So, sir, so you were there for your PhD and your postdoc in the US, and then you had a very nice research environment and a lot of people to talk to, and you would have gained a lot of experience over there. And then what was your driving force to come back to India? Because somebody who would have been there, they would want to continue the career there, and in terms of growth of their career and also. And what is your experience after coming back to India?
Right. So the motivation to come back to India is what you're asking.
Yeah.
I would say that same story. I never planned to go to US, never planned to come to India.
Okay.
Now, of course, when I was finishing up my PhD, I got married.
Okay.
And my wife is a PhD in chemistry, organic chemistry. And in Canada, she was jobless.
Okay.
So she motivated me to come to US, where she was also able to do something.
Okay.
So I think that was the first instance that drove me from Canada to US.
Okay.
And then the second incidence happened again through Nature. That she had sufficient time to look through the Nature journal, and she found advertisement for jobs at IISc.
Okay.
And so she brought that to me, and of course I was not all that keen to do that, but she kept asking me for it.
Okay.
And I applied, and I got the job. And so that was another incidence you can think of where nature has helped me to come to India. Nature
Yeah.
drove me out of India, and nature brought me back to India.
Yeah, nature has played a big role
Yeah.
in your transitions. But just out of curiosity, so how was your research proposal like when you applied to IISc all the way back in 1991, and how has this process evolved over time?
Yeah. So I think that's a good point. As I said that, once I came to MIT, and uh, I think the environment, as Trisha was saying, is very good, fantastic there. I think, there is a difference in the way science is done in Canada and the way it is done in US, and particularly MIT. You could be using the same techniques, same technology, but you would be asking, different questions that matter. So I was working with Raj Bhandari. Raj Bhandari and Khurana sort of have a joint group there, had a joint group there. Uh, and Raj Bhandari is a person who is a sort of a walking encyclopedia. Even those days, not much was available on the computers. Yeah.
True.
So he would be very, very motivating person. So I did, I did publish, several papers in a short time from there. And as I said, soon after that, there was time to come to India. Now, the kind of work that I was doing was, working on initiator tRNA. But during that process, when I was trying to look at the structure function aspects of initiator tRNA, I came across many initiator tRNAs which were non-functional in initiation. Okay. But they were amino acylated well, and that they were formulated also well. So that sort of, gave me an inspiration to think about, is there a way? And this was a genetic method of dealing with it, which genetics I had never done before. Okay. But nonetheless, I thought, if there is a way to, find out how these tRNAs can be made functional, one would know how they interact with the ribosome. And that was really the first thought that I had at that time to put together a project. And so that then allowed me to put together a project where it was a straightforward analysis, what is called as suppressor analysis, that you find out mutations elsewhere in E. coli, which will allow this tRNA to function. As I said that I've never done genetics, the only techniques that I had done in the name of genetics were transformation of bacteria, but transduction, never. And but then, I did write that proposal. And fortunately, that proposal really worked out for us. And of course, the students really work very hard. But then that has been one of the projects which has been continuing in the lab for all these years. We are finding newer and newer questions, and that's keeping us busy. The other project which I wrote at that time was supposed to be something very short-term project, where I knew that this genetics thing I have never done. It might take me a long time to get through something which will be tangible. But another project was on uracil DNA glycosylase, the protein that I worked at Calgary for some time. And the idea there was to develop that as a technology tool. And we did develop that as a technology tool, but we got scooped by, by one of the companies, Gipco, I think it was the BRL. Okay. But then, nonetheless, we managed to publish that paper. But that then led another area of research in the lab, which was on DNA repair. So yes, I think both of these projects were thought before I came to IISc, and both of these have been keeping us busy since then. So I was in a way lucky that whatever I planned at the end of my postdoctoral fellowship at MIT worked. And of course, it did not overlap with the work at my parent lab, because, I have an excellent relationship with my postdoc mentor. And of course, that allows, if I'm doing something different, it allows me to talk to my mentor also. So I think that was something which worked well.
Okay, so you mentioned how the initial the initial concept of your project evolved since you first applied, and has research funding ever played a role in the trajectory of these projects, or in
I would say not really.
steering
I
these
mean,
projects?
there have been occasions where we have ventured into something, different because the funding was available. But I would say that my main research has never been influenced because the funding was available in this area. In fact, I think I would say that in India, it is still something very nice that you can continue to do what you want to do without having to worry about uh, uh, research funding sort of deciding which direction you should go. So if you have a good project, good questions to ask, your projects will get funded. And I guess I was lucky in that that whatever I wrote got funded. And that's really been helping all through my scientific career, that whatever research projects I have written, I guess maybe other than one project, none of the other projects have been turned down. So I think that's been really nice. Yeah, I think it is good in India that way, that your research is not necessarily influenced. Of course, there are opportunities for you to move into different directions, and you can take those directions where there is a lot more funding available. But if you would like to do science for the sake of doing science, and to sort of explore your interest, you can do that in India actually.
That's very
This is
nice.
difficult to do in many other countries. I'm very much aware of it, because I know many people who keep changing their research depending on what kind of funding is available to them.
Yeah, actually.
Yeah, that's a good point, because I've seen professors pivot their research based on the funding.
Yeah. So I think that's definitely a plus point about working in India. Of course, competition is increasing here also. Earlier, you could get a grant funded just for the sake of writing a grant. These days, of course, the money has increased, but not as much as that, one can fund all the grants. So there is some competition, but I would still say that a reasonably good project will get funded.
Oh, okay, sir. So that's very nice that like you have had the opportunity to get most of the research grants funded. So that's a great thing actually. So, and in India also that they offer grants to do research for everything. So now that you have shared about positive aspects of this continuing science in India, what do you think are some of the challenges that you had to face, because you were one of the initial mentors to come to India and start research? So you would have had a lot of challenges, right?
So there are definitely challenges, and I think that one should not be hiding about this, simply because I think one should know what you're getting into if you plan to come to India. Of course, many times good not to know what you're getting into, because you come and then you try to make through it. But I think the difficulties that have been there when I joined in 91, to, let's say, if you were to buy any Sigma product, you are in for at least good 4 months. And this is far better than what people before me joined. You want to get any radioactivity, you are in it for a long time. So you're trying to get any oligonucleotide synthesized, US or any other country. In those days, you call in for a sequence, and within few days you will have the sequence. You call in for the radioactivity, next day it will be on the in the laboratory door before you reach there. So those kind of things are definitely a challenge. Another bigger challenge really was continuous supply of electricity. Even though things have improved a lot, but in those days, you need to really make sure that your reagents are kept in a way that a few hours of power supply failure will not affect your reagents. Things have improved since then, but those were the challenges. Not having any air conditioning in the lab. So I think those were the challenges. Those were challenges because dust will definitely affect your molecular biology experiments, biochemistry experiments. Having non-AC laboratories with a lot of dust coming in, you have to be very careful. Yeah.
Actually, so how did you overcome this? Like, what had you done to make sure?
Right. I mean, so the only thing to overcome these things is, as I said, you plan your experiments in a way that you know that you are going to need the radioactivity. Fortunately, bark was providing radioactivity, but so you need to plan your experiment carefully. You need to finish your reactions so that they don't go bad if you don't complete the reaction. And so there will be times when you will lose your enzymes because the power supply was not there. So yeah, I think one needs to be very careful about those things. And fortunately, there were not way too many power failures. Power failures happened during summertime when the -80s will give up. And then you have to run around and find a functional -80 and move all of your reagents very quickly in 4 or 5 different -80s wherever you can find place. So those were the kind of things you need to do. And I think the students really work very hard. And without students, you really can't sustain any of these things. Now, I'm telling you these things which were there, as you asked me, when I joined. Things have improved since then. We now have generators. So when there is a power failure, generator will come on. Power failure will maybe there for 5 minutes, 10 minutes. Sometimes it is longer but nonetheless, you can manage to run the generator and keep your -80s running. I mean, so for example, you make sure that your -80 is full. And if there is a power failure, and if you don't open it, it will actually sustain a temperature for quite some time.
Actually,
and so that is how really things were kept working.
Yeah, you mentioned about -80, and yeah, I remember one instance here when there was a power failure for half an hour, and there was a panic all across the lab that we might lose our proteins. But yeah, I mean, we survived that. But so now you have talked about your research evolution. You've talked about some of the challenges that you faced, but were there some special Eureka moments in your research, or do you think it was a series of gradual advances?
Right. I mean, I think there is a series of gradual advancement when we do the research. I would say the Eureka moments, I think, are there, and there are plenty of them, I would say, because each PhD thesis gets completed only when a student has a Eureka moment. And so I think that way, I would say there are many. But if I was to sort of nail down the key Eureka moments, I would say that the genetic system that we hypothesized and we thought of when we realized that is working and yielding results, that I think definitely was a Eureka moment, because that set the course of the research quite well. And in fact, we're still busy sorting out some of those things. So I think, yeah, there are always Eureka moments. You need to keep your eyes and your thought process really going all the time. And there are worries many times. There are failures. We chose certain mutants of initiator tRNA non-functional, which did not yield. Then we moved to another non-functional tRNA, which then gave the results. So yeah, I think you need to keep things failures in your stride. Think about why it failed, and then move on to redesign the experiment. And when it functions, when it works, obviously I think it's a Eureka moment, because it really sort of allows you to go along with your hypothesis. I don't know whether if that's what you meant by Eureka moment, or you meant something else.
Yeah, like a flash of brilliance versus like a gradual series of papers.
Yeah.
But yeah, okay. So thank you for sharing all this so far with us. So now you've talked about your own research, but just to get some of your perspective on the Indian research as a whole, and it has come a long way since you first joined as a faculty. Like you mentioned about electricity challenges, and now it's not there so much anymore.
Well,
so
at least in Bangalore and IISc, it is not there that much. But I'm sure in the universities and smaller institutions, these problems are still there.
I see, but okay, but at least it has become much better since you joined, right?
Yeah, and definitely,
grown quite a bit. So, what are some of the challenges that still remain, that we still face in India for the scientific ecosystem, and what could be some of the possible solutions for that?
Yeah, I think one of the major solution really is that you need to choose your problem carefully. And I did not mention it so far to you, but one of the reasons I chose to work with bacteria was that, not that colleagues were not working with tissue culture. My PhD was with mammalian system, and I did work with tissue culture there. But then I think at some point probably had a thought in my mind that, if I have to go back to India, I should choose a system where which I can work well in India. And bacteria doesn't require all that precaution as the mammalian cells do require. And so that was, I think, something that you need to plan ahead of time. You need to plan according to the place that you're going in. Certainly now those restrictions are not there, but there would be other restrictions as to if you want to work on a project which requires you to talk to people in different areas, whether those people are there in that environment or not. Because science has now moved into a phase where working in isolation will not really give you big success. You need to work together with people of different expertise. So that is one thing you have to be thoughtful of. The other thing that you have to be really thoughtful of, and I'm just saying in a random thought manner, is that, when you come to India, you need to work with the students. And remember, not all of these institutions, or not all of the students who come from different institutions are privileged to be trained so well. And so part of your energy will be spent in training the students. In US and many other countries, you will get trained postdocs. So if your idea is to get going very quickly with the trained human resource, then I think you will face a lot of challenge, because the postdoctoral fellows who are good would obviously not remain in India. Of course, there are plenty of schemes that India is now beginning to retain good PhD people to sort of, directly absorb them as a postdoc or at, scientist level. So those are not going to be there in as much as you would want. Most of the postdocs that you will get would in fact require some sort of training through your senior PhD students. So if you're not willing to spend your energy in training a student, I think India will not be a good choice for you to come back. I think you need to be sort of thinking about training the students and driving pleasure out of training students. If you cannot drive pleasure out of training students, I think India is not a place for you. So that is, I think one has to be thoughtful.
Okay. Okay. So now that you have told about, like, one thing is, again, getting a well-trained person in India would be slightly difficult. Now, another question that we would like to ask you is about the existing policies in science. So, for example, now there are a lot of institutes like IISc being made, and a lot of funding is being pumped into IISc, and similar other institute like NITs are there quite a bit. So with all of that, really research has grown, but is money all of it? There could be some policies that needs to be changed. For example, in case of importing animals, like transgenic flies in the case, because I have used flies for my experiments and we have faced a lot of issues getting the flies from Vienna, Bloomington. We have a lot of customs problems, and until India it would come without any trouble, but in India it would stay in a few days, and then by the time we get it, most of the flies would be dead. So hardly anything survives. So all the money that we get actually isn't completely productive. So I think some policies needs to be changed. Also in respect to microscope, a lot of confocals are there, but then if one gets some problem, it takes quite a bit of time for somebody to come and mend it. So other than money, what do you think is needed in the system to be changed? Or
Yeah, I think you've raised very important points, and these are additional challenges to be dealt with when you do research in India.
Mhm.
Some of the things that you have raised, you know, the policies, customs and all these kind of things. Now, I think as the country develops, this is my, personal thinking, I think many of these things will ease out. And the reason I say so is because these custom regulations were really stringent when, I had come here, and even more stringent uh, for people who had come before me.
Okay.
For everything, you needed a permit.
Mhm.
So things have eased out, I think, since very early days. But as you mentioned, I think the same things have happened, with us when we've asked for certain antibodies or something like that, as a gift items, because you don't have regular invoice. If those
Yeah.
flies were being shipped to you as a product or item for which you had an invoice, that would have come out much easier, because they were probably gift to you, and custom people think that you're getting gift in terms of your monetary benefits you are making. They will put you through all kinds of paperwork, which would not happen if they were coming through some sort of, saying that, this costs $10 or $5, and you pay the duty for $5 or $10, and things will clear faster. So I think the part of it is to sort of get around these problems. And getting around these things is really to understand them also, and then alert the person who is sending in these items to you to send it in that particular manner. So many times you can deal with these problems in that manner. But you're right that it is not as simple as if you were to send some item from US to Canada or somewhere, it'll come as a regular thing. No issues, no questions asked. But having said that, there are some countries which are probably worse than India, advanced countries. Say, for example, Australia. You try to send a bacteria or DNA, you've had it.
Okay.
They will ask for a lot of paperwork. But nonetheless, I think things are easing out in India. But it also depends on the political system, which for For example, right now, if you have to place order for anything more than 5 lakhs of rupees, you need to have clearance from the government. This Atmanirbhar thing that they have started, that has also put in a lot of restrictions. So we are hoping that, at some point, many of the institutions will be exempted from that. So these things are, I think, I would say, they come and they go, and one really needs to learn how to get around them.
Okay.
True, I think quite a bit of energy goes into this, but I think this should not be thought of something which will stop your research. I'm sure, I know exactly what you're talking about, because, uh, flies will definitely die. You need to
Exactly.
tell them you put enough of their food in there so that they can live in there. But then if you happen to get them in a airport where there is no AC and the
Exactly.
temperature is 45 degrees,
Delhi.
the flies are gone even with the food.
Yeah.
So yes, those challenges are there. Yeah,
Do
I
you
think
think
those.
implementing people from science into customs, or people who understand a little bit in science, would that help in
I
some way?
don't, I don't think so. I don't think so, because see, this is a bureaucracy,
Mm-hmm.
and when they are given a bunch of rules and regulations that they have to go through,
Mm-hmm.
they may be sort of sympathetic to you, but even they may not be in a position to, sort of overrule too many things. So
Mm-hmm.
basically, I think one needs to understand those rules and then sort of work around them,
Okay.
uh, to get, you know, not get stopped by the customs.
Okay.
But I'm hopeful that in future things will improve, because see,
Huh.
earlier, I'll give you an example. This is an interesting example.
Mm-hmm.
I had a fax machine, that paper, where you
Yeah.
transmit certain thing. And so that I had at home,
Mm-hmm.
when I was at MIT. I brought it along with me. Oh boy, you wouldn't believe how tough time I had to get it, because that was a banned item.
Oh my God.
It took 5 or 6 months to get the license, to get it in there. And by the time I got it, it was just outdated. And I ended up spending a lot of money to clear that. So I think that was totally unthoughtful of me to do that. I should have just left it there. Would have been better off. So not knowing certain things is also a problem.
Okay. Okay, so now I think we have discussed quite a bit about challenges in different ways. Now, according to you, what do you think the success of a lab? Like, do you think that the amount of money or grants the lab get, or is it in terms of publishing, number of publication, or a high impact journal, or the basic question that one has in mind? So, what is your perspective on that? Like
I think it is a bit of
success.
everything. I wouldn't say that is only just one of the things, bit of everything. But I think the biggest thing that I consider a success of the lab is really, um, if you have thought of something, you have been successful in, in certain, you propose certain hypothesis, and it yielded to your thought. I think that is a success. And more of these hypotheses you keep proposing based on certain, uh, background knowledge that you have, and you get more of those answered, uh, I would call it as a success. Now, to do these things, certainly you need money. Certainly you need other things that you mentioned.
Yeah.
To get money, you need to publish. So they're all sort of interrelated. So that's the reason I said that it's not just one of these things. I think it's a sort of, bit of everything that needs to go along for you to call yourself a successful person in terms of doing science. So, I think somebody who wants to think about coming back to India, they should split their success in two compartments. One as training, another one doing research. So you get success in one part, you can tolerate some failures in the other part. And so that will keep you going. So think about doing compartments. One compartment, definitely, if you are motivated to train a person, you will be successful. There's no doubt about that. Other one, depending on many other factors, if things go well, you will be successful. And if you try hard, definitely you will be successful. So I think that's a sort of sort of idea that I had when I came here.
Okay.
That if I want to be successful, I should at least think of some things that I will do well, and I'll feel good about it. So if I can teach students, some courses, some, training, I will feel good about it. And if I feel good about at least one part, I will do well in the other part. But if I just think of research and I'm not, getting anywhere, I will be discouraged from doing research, and that will then take me downhill. So, one needs to plan things in that manner that, the balance of the thing is something that you feel good about.
Okay.
So basically, success is about how rewarding the work that you're doing is for you.
Yes. And how good do you feel about what you're doing?
Right. Yeah.
Feel good about what you're doing, I think it is success. You need to see it from your perspective.
Right. Yeah, that's a very good point.
Yeah.
And so you have just a quick transition to mentorship, for example. So you have mentored a lot of students in your career so far. And so what has been your philosophy like when you're mentoring grad students and postdocs?
Yeah, so my philosophy is, I don't have a very fixed philosophy. The reason I say so is because every person who joins the lab is different, somewhat different. Yeah. And the way you need to then sort of communicate with them, to relate with them, has to be slightly different. Has to be slightly different also because the places that they come from. A lot of people come from cities, a lot of people come from village background. And so you need to sort of, talk to them at a kind of wavelength that they can feel comfortable with. That's number one. Number two, really, I would say, is that if I can make them own their projects, I would consider myself very successful as a mentor. Once they own the project, then there is this constant discussion. When they are thinking of some problem, they are thinking of some question in that, you discuss it. You design the experiment. Again, you discuss the design of the experiment. And then they will feel motivated because, there has been their contribution in that experimental design. If I just tell them continuously, okay, you do this, you do this, you do this, I'm just kind of as if, I'm giving them, handing down experiments after the experiments, and I'm constantly monitoring them. Why have you not finished this experiment? Why have you not finished this experiment? That will not motivate them at all. They will be bored very quickly. But once they start owning the problem and they start discussing the problem, they will on their own do the things with a lot more motivation, with a lot more ownership, because they're doing things for themselves, not for me. And if I keep telling them, do this, do this, do this, then they're doing things for me, and that is not something they will be very happy about. So I think these are the things that one needs to keep in mind while you are mentoring, whether it is a student or a postdoc or anybody for that matter.
I see. So this is basically essentially what you talked about. Feeling successful in research is related to being rewarded by the work that you do, and the philosophy is essentially sharing that feeling with your students as well.
They feel excited about it.
Right.
If they can add one step to the methodology or the way we will approach it, they will feel good about it, because then there is their contribution in it.
Right. And so do you have any advice for all the grad students and postdocs who would be listening to this conversation, both in India as well as abroad, to build their career?
I think the only advice I will have really is that there are clearly no shortcuts. one has to really work hard and take pride in what you do and make the best of what you have. That is the only advice I will give, because I think I have followed that. Take pride in what you do the best you can on whatever you're working with. There are always, the grass on the other side of the fence is always greener. And that should not disturb. If you're doing a project, if I'm working on bacteria, I'm fine. I don't worry if, it is some sort of, not that fashionable research. So long I can motivate my students, so long I can feel excited about what I'm doing, I think that is what is more important. So I think hard work, take pride in what you do, and just really, do the best you can. That's all really my advice would be.
That's great, sir. That's great advice, actually. I think it's applicable for everyone in any time in their life. I mean, it's not only the students who are in India, abroad, or doing science. I think it's a life philosophy that you have given. But that's indeed very great. Thank you. Thank you so much, sir. So thank you for giving us your time, and we feel really privileged and for sharing all your journey so far and all your valuable experiences. It's been very nice talking to you, and thank you so much. Thank you.
Thank you. It's been a pleasure. Yeah.